Hello, everyone. I'm Dr. Matt Bochenek. I'm a senior research fellow in the Dr. Anderson and Dr. Linger labs at MIT. And so today, I wanted to describe some of the work that we've been doing developing anti-fibrotic materials for the use in immunoprotection of cell-based therapies. So a little introduction into immunoisolation devices. So what these are, and the whole goal is to protect therapeutic cells within a semi-permeable membrane. So they're protected from the immune system, but they can still function to secrete some sort of therapeutic protein that can regulate a disease. So small molecules and things that the cells need to survive, like oxygen, glucose, can easily diffuse in. The cells can produce that therapeutic protein of interest, which they'll secrete, which will diffuse out of the device to then affect the response to control the disease. However, this membrane blocks entrance of important immune cells like T cells that would otherwise reject these cells. So really what we're trying to do is create this living drug factory where these cells can continuously produce this drug of choice. So we don't need to repeat injections or oral doses. And most importantly, we won't have to give immunosuppressive drugs, which have a lot of negative health risks. You're more susceptible to blood lymphomas or other types of infections. And so for these types of devices, you can use multiple kind of cell types. You can use primary cells that are isolated from donor organs. You can use cell lines that have been engineered to secrete some sort of therapeutic protein of choice, or you can even use differentiated stem cells as a solution to this need for or shortage of donor organs. They can come in a variety of shapes, so they can be spherical, like microspheres. They can be encapsulated in planar sheets, or they can even come in these cylindrical fibers that contain your cells of choice. So particularly, I've been interested in a beta cell replacement for the treatment of type 1 diabetes. So in type 1 diabetes, these patients have an autoimmune response to their beta cells, which ends up destroying these cells, so they can no longer produce insulin. And so the current standard of care for these types of patients is they break their finger, they monitor their blood glucose levels, so either manually inject the amount of insulin that they're needed, or there have been advances where you can get continuous glucose monitors and even insulin pumps. However, it's been very difficult to fully recapitulate the dynamic kind of glycemic control that's normally seen in the body. So normally this is blood glucose homeostasis is maintained by the islets of Langerhans, which are cell clusters that secrete various hormones, such as insulin, which will lower blood glucose levels, and glucagon, which will raise blood glucose levels, and then somatostatin and some other hormones that tight glycemic control. And so the body has evolved, and these islet cells have really evolved to maintain this tightly regulated blood glucose levels, which you can see within this narrow range that is highlighted by the red dashed lines. And using a continuous glucose monitor of patients that have undergoing manual insulin injections, you can see that they're often outside of this range. And even when you use an insulin pump that can try to precisely time when to inject this insulin, they're still often out of this range. And this is really being outside of these ranges is what leads to the long-term complications and the increased morbidity associated with type 1 diabetes. And so I've really been focused on how can we replace this cellular control system to really restore this physiological glucose control. And so my previous lab, I worked in a phase 3 clinical islet transplantation where we would take a donor pancreas, we would enzymatically and mechanically digest the organ into small tissue fragments. We would then isolate the islets and infuse these into the liver of type 1 diabetic patients, where they become lodged in small capillaries, and then these cells would just live in the patient's livers. And so this was very effective, and it would restore the dynamic and that physiological blood glucose control. And we had some patients that were insulin independent for over 10 years. However, there are some drawbacks. So, like I said, this requires systemic immunosuppression and those drawbacks. And there's also a limited number of organs that can treat all the diabetics. And so I've been working on kind of like a simple method of cell encapsulation within alginate microbeads, where the islets can become entrapped within these hydrogel spheres. And alginate is nice because it's very cell-friendly, and it cross-links in the presence of divalent cations to solidify this gel around the islets. And so this strategy has worked very well in many diabetic mouse models. So here, this red dashed line, any blood glucose level below this line is within the normal range, signifying that these animals are no longer diabetic. And these encapsulated islets can really function for very long term, up to a year in these mice. And this has led to a small number of clinical trials with encapsulated human islets and these same kind of alginate-based formulations that were tested in these mice. However, the human patients only experience transient islet graft function and very little therapeutic benefit. And so one of the main issues is that investigators reported clumping of the microcapsules, adherence to a mental tissue, and a strong fibrotic response of these islet grafts. And so in here, you can see this thick layer of fibrosis surrounding the capsule. In this kind of dense ECM and cellular network, it really restricts the diffusion of oxygen and other nutrients that are important for the cell survival and ends up killing these islets. So kind of historically, people have debated, is this an islet problem or is this a material problem? So isolated islets can vary in quality depending on the isolation process, which is very artisanal, and also just the organ donor and like that, the quality of kind of how they happen to pass away and some of the issues surrounding the organs. And then another potential cause for this lack of function is that this intraperitoneal transplantation site in the abdominal cavity has been associated with low oxygen levels, which may not be ideal, and maybe this led to this graft failure. However, in certain rodent models, people have shown that there is a foreign body response to alginate-based formulations, and this has a consequent negative effect on islet graft function. And so we wanted to test this. So we took those same formulations that were implemented in clinical trials. We formulated them as empty beads, so not including any islets, and implanted them into a higher order animal species, non-human primate, that perhaps is more similar to a human patient. And so by 1 week, we started to see cells attached to the surface of the microbeads, and by 4 weeks, these were completely embedded in fibrotic tissue. So very similar to the reports with the encapsulated islets. We looked at the different cell populations that were responding to these alginate-based formulations and found CD11b myeloid, foreign body giant cells, apple smooth muscle, actin, myofibroblasts, and many of the cell types that are associated with foreign body responses to implantable materials. And so these results really suggested that the fundamental problem was material-based and not necessarily with the islets themselves. So a very talented postdoc, Arturo Vegas, at MIT, was screening small molecule modifications to the alginate backbone to find alginate derivatives that could inhibit this fibrosis or this foreign body response to materials. So of these 800, he found 3 different formulations that could prevent fibrosis in mice. And then these were shipped to Chicago, where we could then test these in the NHP model as we had tested the clinical alginates. And so from these 3 formulations, we found 2 that were the most effective at reducing fibrosis in the alginate spheres. So then we wanted to see how this would protect allogeneic islets. However, over the course of these experiments, we observed that we would transplant these spheres near the liver and the abdominal cavity. However, over time, they tended to descend and settle within the Douglas space in the base of the pelvis. And so we really didn't want to form any clumps, because if you can think that any cells in the center of these clumps are going to be super far away from any blood vessels, and that is going to restrict, basically similar to fibrosis, and lead to loss of viability. So we hypothesized that we could place the encapsulated islets within the bursa omentalis. So this is this thin membrane that tissue bilayer, basically, that hangs like an apron above your intestines. And so we hypothesized that the friction of these omental kind of folds and these tissue layers will prevent the sedimentation of these alginate spheres. Transplantation technique. So here is a video of the transplantation technique. So laparoscopically, we make a small incision in the greater omentum. Then our encapsulated islets and spheres are within a syringe, and then they are infused laparoscopically into the bursa omentalis. And so here you can see that the encapsulated islets are within that tissue bilayer. So it's a little hard to see, but you can see maybe like one capsule where it kind of goes underneath that fat tissue, and it's kind of in between that tissue bilayer. So then after 4 weeks, we wanted to retrieve the islets first to observe how they look within the intraperitoneal space. And so we can see that they are well distributed within that tissue bilayer in the bursa omentalis. They're also translucent, which is always a good sign, so that indicates that there's no fibrosis of these spheres, since you can still see through the capsules. And so then he also infused a large amount of saline and is going to aspirate the solution shortly. And so this was to see if any of the spheres had leaked out of this bursa omentalis, or this kind of tissue membrane. And so you can see we didn't find any spheres within that aspirated fluid. So then we wanted to look at the biocompatibility of the spheres after retrieval. And we found very little fibrosis in 6 out of 7 of the NHP that were transplanted. So these are images of inverted phase contrast. So any kind of dark sphere indicates that that sphere was basically completely covered with fibrosis. However, we found that only occurred in one of the NHP that was retrieved at 4 months. Then we took these retrieved spheres, we dissociated any cells on the surface and quantified the number of CD68, CD11D positive macrophage cells, which are important kind of marker, important cell type that's involved in the foreign body response to implantable materials. And we can find a significantly reduced number of these cells compared to even a plain alginate control that contain no allogeneic islets inside. Then we estimated the number of viable cells per islet throughout the course of the experiment from pre-encapsulation all the way up to the 4-month retrieved islets. And we find that ZY115 can protect viable islets at similar, comparatively high viability compared to even before transplantation. Then we stained the retrieved islets with diphazone, which is a red dye that binds to zinc within mature insulin granules, and we can see bright red staining indicating mature or engrafted mature endocrine tissue. We then performed a glucose-stimulated insulin secretion assay where we incubate the retrieved islets in a low glucose solution for 1 hour and a high glucose solution for 1 hour, and we measure the amount of insulin being secreted into the supernatant, and we can find a significantly increased amount of insulin under high glucose compared to low glucose, indicating that the islets are maintaining their glucose responsiveness. Then we used a microfluidic-based parafusion assay to probe some of the intracellular coupling factors that are important in the insulin secretion pathway. So using a set of dyes, we are able to monitor basically the intracellular calcium levels and then the mitochondrial potential, which are important pathways or in the insulin secretion pathway. So if you look at the center figure, maybe I'll go into detail about how this pathway works. So you have glucose come in, it gets metabolized by the mitochondria and converted to ATP. This closes potassium ion dependent or ATP dependent potassium ion channels. This leads to a buildup of potassium ions within the cell that depolarizes the membrane. Then this opens up voltage dependent calcium ion channels, which causes an influx of calcium. And then that influx of calcium causes mature insulin granules to bind with the cell membrane for insulin exocytosis. And so here on the top right, you can see that once we stimulate with a high glucose at 5 minutes, we get a spike of intracellular calcium levels that is not significantly different than pre-transplant levels, indicating that these voltage-dependent ion channels are intact within the retrieved islets. And similarly, we see a similar decrease in the mitochondrial potential, indicating that these mitochondria are actively engaging in glycolysis and important for in the insulin secretion pathway. So in this study, we can show that the long-term protection of viable and glucose-responsive allogeneic islets was achieved in non-human primates without any immunosuppression. And from these and some other studies that we've performed, we have found 2 small molecules that resist fibrosis, and we can graft these onto a variety of materials, and they have a similar effect. And these results initiated the formation of a startup company, Sigalong Therapeutics in 2016. And they just have recently transplanted the first patient with an encapsulated engineered cell product that secretes factor VIII for hemophilia A, which is exciting to see how this material functions in human patients. So I'd like to thank my advisors, Dan and Bob, my grad school advisor, Jose Oberholzer, all the other graduate students, postdocs, technicians that have helped for these projects. And yeah, normally I would take any questions, but